In the last session, we had stopped at the molecular mechanism of Auxin action. So, I will start from here itself. We already saw that there are three regions where we have the receptor proteins for the auxins, and one such kind is ABP1, which is generally seen in the endoplasmic reticulum. Now, once the auxin bind to the receptor, the first change what we can realize is the increase in the membrane voltage. And when they have experimented with antibodies, they have seen that even the binding of antibody, they have developed the antibody for ABP1. So, once that antibody comes and bind to that, it can bring about the effect of auxin by bringing about the hyperpolarization of tobacco mesophyll protoplast, and that is where they experimented. And they have also seen that it can bring about the stimulation of protein extrusion. So, not necessarily that auxin binding has to take place, but an antibody which has developed against the auxin, if it can bind to the receptor, the same effect of auxin binding can happen. Now, not only the binding phenomena is there in the auxin action, you can see after the binding of the auxin to the receptor protein, there are certain signaling intermediates being involved before the gene action, and one such signaling intermediate is CDK, that is cyclin dependent protein kinase, and another one important one is MAP kinases, and that is mitogen activated protein kinases, and both are involved in phosphorylation, and these can basically involved in the cell cycle or a division of the cell and enhancement in the cell growth. So, these two signaling intermediates are basically involved in the auxin action, and together with that you can see there is a rise in the level of calcium, and it has been found that when 2,4-D was applied to the plants, there can be increase in the calcium levels up to 280 to 380 nanomole level. And auxin decreases cytosolic pH by 0.2 units, which in turn can promote the activity of ATPase. So, on binding of the auxin to the receptor, there is a signaling intermediate getting activated. As a result of that, you can see that there is a decrease in the cytosolic pH. And not only these intermediates are involved, you can also see that auxin can stimulate phospholipase A2, in short we call it as PLA2. And by the action of this enzyme, fatty acids are released from the membrane phospholipids, and which can serve as a substrate for lipoxygenase. So, on the action of auxin, phospholipase A2 gets activated, which will release the fatty acids from the membrane phospholipids, which will act as the substrate for lipoxygenase. And the other product of the reaction, together with this particular reaction, you can also get another product which is called as lysophosphatidyl-choline, and which can activate a protein kinase, and that can bring about a phosphorylation phenomena. So, we have seen that CDKs are getting activated, which is bringing about phosphorylation phenomena. MAPKs, mitogen activated protein kinases, are getting acting as signaling intermediate, again can bring about phosphorylation. We have seen that PLA2, auxin is stimulating PLA2, that is phospholipase, which releases the fatty acids, and as a result of that membrane phospholipid fatty acids can act on or can act as substrate for lipoxygenase. The other product of this reaction, that is lysophosphatidyl-choline, can activate certain protein kinases, which again can bring about phosphorylation. So, this all phenomena can finally bring about the alteration in gene expression. And if you try to see that once the auxin is applied, there is rapid changes in the levels of translatable mRNA, and the whole class of mRNA is divided into two classes, or the entire mRNA which is getting the levels of mRNA which is getting enhanced can be divided into two basic classes. One is primary responsive genes, and the second is secondary responsive genes. If you try to see that the primary responsive genes which are getting activated is the one encoding proteins that regulate transcription of secondary response genes. So, the primary responsive genes, immediately after the auxin application, a set of genes get activated, which we call it as primary responsive genes, and the end product of that is are certain transcription factors which can activate secondary responsive genes. And the product of these primary responsive genes are also involved in cell-cell signaling. They are also involved in stress adaptation. So, basically the primary responsive genes which is getting activated immediately by auxin action is bringing about three effects. One is encoding proteins that regulates transcription of secondary responsive genes. Second is involved in cell-cell signaling. Third, they are involved in stress adaptation. There are basically 5 major class of auxin responsive genes. You can see the list over here. That is auxin IAA gene family. Second is SORE, that is small auxin upregulated RNAs. Third is GH3. Fourth is ACC synthase. Fifth is glutathione S transferase. In short, we call it as GSTs. So, they are basically a class of proteins stimulated by the stress conditions. So, there are basically 5 major class of early auxin responsive genes, and the end result or end product of that may get activate the secondary responsive genes. So, this is all what we are describing in the case of auxins. So, to conclude the topic of auxins, let me tell you, we started with some historical aspects or experimental aspects which led to the discovery of auxin. We went through the portion wherein we learned about how the biosynthesis of auxin takes place, the different pathways of auxin biosynthesis taking place. Then we moved into the important physiological functions, and we studied in detail about the cell elongation taking place, the phototropism taking place, the gravitropism taking place. Then finally we saw how the auxin application will result in the gene action, expression of certain genes happening. So, this is what we have learned in the case of auxins. Now, we move into the second important plant growth hormone that is cytokinins. Again, we will be describing certain historical aspects or experimental aspects which led to the discovery of cytokinins. In 1913, Heberland in Austria, he saw that the vascular tissue is containing certain water soluble substance which can stimulate the cell division in potato. So, he took certain water soluble substance from the vascular tissue, and that was applied to the potato cells, and he saw that potato cells are dividing, it is getting proliferated. And similarly, the same action was further studied in many other natural products such as yeast extract. It was also studied in tomato juice. It was studied in coconut milk, and these natural substances also have the capacity to bring about the cell division phenomena. And now we know that in coconut milk, the natural cytokinin what is present is creatine. And another natural cytokinin what was discovered was kinetin, and this is the first cytokinin which was discovered, and it was discovered as a DNA breakdown product. Once the DNAs can be broken down into simpler compounds, you get kinetin as a product. And now we know that adenine, which is an aminopurine, has got a promotive effect. Also, we have seen that autoclaved sperm DNA has a cytokinin effect. And Scooge and Miller, they first fractionated the heat treated DNA and obtained kinetin from it. And also creatine, already we saw that it is one of an abundant and natural cytokinin, and it is extracted from immature endosperm of CMAs. and it has got two different forms, one cis form and trans form. These are both isomerases of same thing. And if you try to see that artificial forms of cytokinin, we can see that they are basically N6 substituted aminopurines. The common artificial cytokinins are benzyl aminopurine, in short we call it as BAP, and diphenylurea, and both are N6 substituted aminopurines. And if you check the structure of a cytokinin, you can see this is the trans, towards the left you can see there is a trans cysteine, and this isomeric form is the cis cysteine. And coming to one of the topic with regarding to the bioassay, if you want to analyze the cytokinin action in plant tissue, the basically what you can perform is assay is called referred to as bioassay. You can take the particular plant tissue. For example, tobacco pith is one of the best tissue wherein you can perform the bioassay. You can see that the cell proliferation will be taking place extensively. The amount of cytokinin present in the tobacco pith itself can bring about the proliferation of the cells. And if you add more of cytokinin externally, it can still further grow or enlarge, the proliferation of the cells can take place. Another cell tissue, plant tissue which can be taken for the performance of bioassay is the quaternary leaves. The quaternary leaves contains a certain amount of cytokinin, and once it is cultured artificially, the quaternary leaves expand. And based on the expansion of the tissue, you can assess the quantity of cytokinin what is present in the quaternary leaves naturally. And obviously, if you give more of cytokinin externally in the medium, you can see further proliferation or further expansion of the quaternary leaves. So, these are certain tissues which can be taken as for assaying the quantifying the level of cytokinin present in the tissues, or even which can be supplied externally, and this we refer to as bioassay. So, it is the application of a biomaterial for assaying the particular content present with the tissue, and here we are assaying the content of cytokinin which is present in the tissue. If you check the cytokinin in the presence in the plants, you can see there are free form as well as bound forms. The free forms are what you see over here: zeatin, dihydrozeatin, and isopentenyl adenine. These are generally free forms, and the free forms can bring about cytokinin activity. But together with the free forms, you can also see bound forms. Bound forms are cytokinin which is complex or coupled along with certain other compounds. For example, plant tRNAs contain zeatin as hypermodified base. So, here zeatin is along with the tRNA. This is a bound form, it is not free form. What is the significance of this? The significance is that only when zeatin is free, it can bring about the cytokinin action, or when it is in bound form, it cannot have a cytokinin action. So, whenever the significance of this is that cytokinin generally when it is being transported, not only cytokinin, any other hormone, whenever it is transported to any other from the site of synthesis to site of action, it is generally transported as in a bound form. Why? Because on the way it will bring about the cytokinin activity if it is in free form. So, generally it is transported in bound form. And when will it become free? It will become in free form once it reaches the site of action. The bound form will get separated and the cytokinin can act freely. So, this is the significance of free form as well as the bound form, and this is a general case which you can see in the case of all growth hormones, because on the way of transportation it has to be in the bound form, otherwise it will bring about action on the way itself. So, in order to prevent that, it is transported in a bound form, and once it reaches the site of action, it is being freed so that it can bring about the physiological function. Now coming to the biosynthesis of cytokinin, you can see that isopentenyl pyrophosphate combines with AMP, adenosine monophosphate, and results in the synthesis of isopentenyl adenine ribotide. So IPP, in short we call this as IPP, isopentenyl pyrophosphate, combines with adenosine monophosphate and gets the formation of isopentenyl adenine ribotide takes place. And by the action of the enzyme cytokinin synthase, we get the synthesis of isopentenyl adenine or creatine or dihydroxyatine. So these are the natural forms of different cytokinins. So this is the general biosynthesis process taking place within the tissues. And once synthesis has taken place, generally it has to be transported from the site of formation to site of action. So root to shoot, generally transportation takes place via xylem along with water and minerals. In the case of auxin, we have already seen it was through phloem, but in this case it gets transported via xylem along with the water and minerals. The major site of synthesis are generally shoot and root meristems, and also young maize embryos, young developing leaves. These are the main sites of cytokinin action. Transported as creatine ribosides, once they reach the leaves, they are freed. I already told you what is the significance of this coupled form or conjugated forms which are being transported. So generally the transportation of the creatine takes place in a riboside form, and once they reach the leaf where they have to act upon, they are generally freed. A signal from the shoot can regulate the cytokinin transport from the root. Already we have seen root is one of the best location of synthesis of cytokinin. And what makes the transportation of the cytokinin from the root to the shoot? Generally, if there is a requirement of cytokinin in the shoot, a signal is coming from the shoot to the root region, and accordingly it moves, the cytokinin is being transported from the root region to the shoot region. And cytokinins are rapidly metabolized by plant tissues. Once the cytokinin bases, I already told the conjugated forms or coupled forms, either it will be in the nucleotide form or glucoside forms. Once it reaches the site of action, it can be freed. One of the method is that glucosidases act on that. If it is glucosidic forms, glucosidases act on that, and the cytokinin is freed, so that again it returns back to the active form. So cytokinin getting converted into nucleotidic or glucosidic forms, which is inactive, generally used for storage, and once it is transported from the storage region to the site of action, there it gets active again by the action of glucosidases. So the glucosides are removed from that, and it becomes active again. Now we go into the different biological roles played by cytokinins. One is that shoot apical meristems of cytokinin overproducing plants produce more leaves, and secondly you can see more chlorophyll is synthesized. If you apply cytokinin, there is synthesis of more chlorophyll. Adventitious roots from leaf veins and petioles occur by the application of cytokinin. Leaf senescence is retarded, apical dominance is reduced. Cytokinin overproducing plants are generally stunted and shortened internodes. They have very small internodes, so they have very less in length, they grow in less in length. Rooting of stem cuttings are also occurring because of the application of cytokinin. Auxin and cytokinin ratio is regulating the morphogenesis in cultured tissues. In tissue culture, you know that basically the auxin and cytokinin ratio is regulating the morphogenesis, that is the development of roots or shoots from the cultured tissues. It can modify apical dominance and promote lateral bud growth. And auxin cytokinin can regulate the plant cell cycle, that division of the cell cycle, and based on which the cell tissue enlarges. and the basic role is played by the action of cytokinins. CDKs is activated, and CDKs, you know, is cyclin dependent kinases. So here the kinases will ensure the phosphorylation, and based on that the cell cycle is being regulated. The application of cytokinin can delay the senescence, and another main important biological role is that it can promote nutrient mobilization, and another important function is it can promote chloroplast maturation. Here you can see that when you keep the proplastids in dark, it is generally getting converted into etioplast. So chloroplast development will not take place. And when does chloroplast development takes place from proplastids? It is based on the light falling to that, proplastids get converted into chloroplast. If it is kept in dark, you can see proplastids only get converted into etioplast. But these etioplasts can be converted or can be transformed into a mature chloroplast by the action of cytokinin. No involvement of light is required, but only cytokinin, action of cytokinin is required even if it is dark. So that means similar to that of light, cytokinin has the capacity to convert the etioplast into a mature chloroplast with extensive grana, with more chlorophyll and more photosynthetic enzymes. So it is basically promoting the chloroplast maturation. And you can see that it is promoting cell expansion in leaves and cotyledons. And you already seen that Auxin has got the capacity to bring about cell elongation. There you have seen that it is basically because of the activation of expansins, activation of hydrolytic enzymes, activation of buildup of osmoticum, and activation of fresh synthesis of cell walls. And here the promotion of cell wall expansion in leaves and cotyledons is basically is due to the increase in mechanical extensibility of the cell walls. The cell walls has a special capacity to be elastic in nature, extensibility, just like the elasticity, and that is happening as a result of the application of the cytokinin. And this is not accompanied by proton extrusion as in the case of Auxin. There you can see that in the case of Auxin, the first phenomena is the proton extrusion, and that is followed by the activation of expansins, buildup of osmoticum. Similarly, the hydrolytic enzymes are getting activated. So it is all followed with the proton extrusion. But here you can see there is no proton extrusion, but still cell wall extensibility takes place because of the action of cytokinin. And it can another one of the biological role is that inhibition of cell elongation in stems and roots. Although it will promote cell expansion, the cell elongation phenomena is basically inhibited by cytokinin in the case of both stems and roots. Here there are some cases wherein you can see the clear-cut action of cytokinin. Here tobacco plants are overexpressing the gene for cytokinin oxidase, and shoot growth is strongly inhibited in the transgenics. So here you can see transgenics are overproducing cytokinin oxidase, and based on that the tobacco plants which are overexpressing this enzyme cytokinin oxidase will result in inhibition of the shoot growth, and this is a wild growing normal plant, but here the cytokinin oxidase is being overproduced in this case. So inhibition of growth takes place, and in this case you can cytokinin suppresses the growth of roots. If there is more of cytokinin, it can suppress the root growth, but here the root growth profusely takes place. And here you can see cytokinin suppresses the size and cell division activity of roots. The A is a wild type and B is the transgenic, and here cytokinin suppresses the size and cell division activity of roots. Here because of the cytokinin action, it is suppressing the size and cell division. That is why you do not see more of thickness of the roots. But here in that transgenic type, you can see there is no suppression of the size and cell division is taking place. As a result of that, there is a thickening of the root occurring over here. The auxin and cytokinin ratio is regulating the morphogenesis in cultured tissues. Already we discussed in the heading biological roles of cytokinin, and there you can see that the morphogenetic development of the cultured tissues, the development of the roots, the development of the shoots, all take place with the difference or variation in the auxin and the cytokinin ratio. Here you can see that leaf senescence is retarded in a transgenic plant containing cytokinin biosynthesis gene. This is a case wherein IPT gene is being introduced. As a result of that, more cytokinin biosynthesis takes place, and the plant expressing the IPT gene remains green. Why? More chlorophyll synthesis, more maturation of chloroplast takes place, and less degradation of chlorophyll takes place, and the plant remains green. But in the other case, you can see the age matched control advanced senescence, and no photosynthesis is taking place because less of cytokinin is produced, and no IPT gene is here in this case. Also in this case you can see that this is an experiment with the radish cotyledons, and the effect of cytokinin on the expansion of radish cotyledons takes place. If there is light, there is a lot of chlorophyll synthesis, but there is less expansion. But on application of C18, you can see more of expansion of the cotyledons takes place, and that is because of the cell wall extensibility. Here also, even if it is in dark, the expansion of the cotyledons takes place along with the application of C18 because of the cell wall extensibility. The effect of cytokinin is bringing about the expansion of the cell because of the cell wall extensibility. And now we move into the next topic, that is the mode of action, how the cytokinin is bringing about the action on application, and that we will be seeing in the next session, and we will stop here the session, and we will continue with the mode of action of cytokinins in the next session.